So before we introduce Dr. Bjornson, I wanted to make a big announcement. We have our 99th and 100th registry in Rare X. So Christina and I tend to be a little competitive. And we knew we would make it by the end of the conference. So I think the next 100 are going to be a little bit harder, but with all of your help and encouraging people, having those reports just to take to all of our doctors in of itself is a huge piece of the Rare X program. So without further ado, I want to introduce our major WSS champion, Dr. Hans Bjornson, who just flew in from the ASHG conference in the middle of the night last night and is headed to Iceland today. So if he stumbles over any words, there's a reason. Dr. Bjornson is a professor of translational medicine and pediatrics at the University of Iceland. He is also an associate professor of genetics at Johns Hopkins here in Baltimore. He founded the first epigenetics and chromatin clinic at Hopkins in 2012 and has significant clinical and research experience with WSS. And we are so lucky to have him on board, the most humble man you will ever meet. And he has been an incredible resource and so generous with his time with our community. So without further ado, welcome Dr. Bjornson.
Thank you so much for inviting me and having us. It's so nice to see how this has evolved from the first conference to this. It's just so impressive to see the progress. And I'm just thrilled to get a chance to tell you a little bit about the science. I'm not going to talk about what you've heard from Tinna. I'm not going to talk about it again. You heard that talk. But you know, I know this is a family conference, but I wanted to share some of the ideas and the data that we have and how we think this could yield kind of therapeutic strategies kind of going forward. So I always start with this. You know, we've had the human genome, which you can think about as a book. You know, it's a book that we have. The genome is kind of the information for everything that the cells do. And one of the challenges that book has is it needs to be read in many different ways. So, you know, because there are many different cell types and some of them are very different. If you think about a sperm cell or a neuron, they have different components. They need to read the book differently. And one way to read a text differently is to have highlighters, something to pick out the pieces that you need for that cell type. And it would be great if you know, one way would be if each system had one highlighter, and this thing called the epigenetic machinery. It sounds like something from science fiction or something, but one way to think about it is to think about it as the genome's highlighting system. It's a way to kind of pick out the pieces that are needed in different cells and make sure that other genes behave in the right way. And so what are these epigenetic marks? These are little additions to the DNA or the histones, and they kind of help the cells know how to behave. And the reason why we're excited about these marks is that they're reversible to some extent, unlike the DNA, and they're known to be affected by the environment. And there are kind of two big categories. There's something called DNA methylation, and then there's something else called histone tail modifications, which is super important for Wiedemann-Steiner. So DNA is not floating around. It's packaged in something called the nucleosome. And the nucleosome is basically a wrapping of DNA. And then there are these little tails that stick out, you know, on either side. And then, you know, the machinery can put on modifications on these tails, and that dictates whether the DNA is active or not active. And the mark that matters for Wiedemann-Steiner is this thing called H3K4. That's a mark that protein puts on. So it's an active mark. It's a way to open up chromatin. And And there are many disorders that we now know where the machinery to maintain these marks is broken. So it's unable to function. And I've spent a lot of time working on this related disorder called Kabuki syndrome, and that has yielded a clinical trial. Doesn't mean that that will work, but we're at least at the step where we can test things in patients. So I'm going to tell you a little bit about that, and I'm going to tell you about some of the differences that we find between Wiedemann-Steiner and Kabuki, and why we think kind of thinking about this group as a whole might help us kind of get to answers faster. So like Libby said, we hosted the first conference, and I'm grateful to work with multiple people like Dr. Harris, Dr. Farner, and Dr. Ng at that clinic. And our idea was, these disorders are rare. And when we started this, there was only 20 disorders that were known, and there's more than 90 at this point. And by seeing them in one center and building excellence, we could kind of learn some kind of general principles. And I'm going to start out by this success story from Kabuki syndrome. So this is also a disorder of histomethylation, just like Wiedemann-Steiner. And what was amazing about this disorder when it came out about 12 years ago, that the two causes, the factors that maintain that if you lose these factors, you get this disorder called Kabuki syndrome. They both affect the ability to open up chromatin. So the first one is called KMT2D. It's very similar to the KMT2A, which is mutated in Wiedemann-Steiner. And then the second cause of Kabuki syndrome is something called KDM6A. And both of it kind of led to an inability to open up chromatin. So we thought, hey, that's a clue. That's a clue that we can follow, and maybe we can yield some therapeutic strategy based on that. And to do that, you often need to have mice. So we developed the first mouse model for Kabuki syndrome, similar to what Tinna told you about, that we're now doing for Wiedemann-Steiner. The mice had a mutation that was similar to what was seen in patients with Kabuki syndrome. And they had many of the same phenotypes. They had growth retardation, so they didn't grow very well, these Kabuki mice. And they had the same immune problems as we see in our patients. So we felt convinced that, look, these mice, can model what we see in the patients. We've also found some new phenotypes in these mice, which we think are clues to kind of keep pushing forward. One surprising thing in Kabuki syndrome was that the bones are shorter in the mice, just in individuals with Kabuki syndrome, but the growth plate itself is big. And again, we didn't expect that, and that's something that we're actively exploring. Kabuki, just like Wiedemann-Steiner, has a lot of gut issues, a lot of failure to thrive, diarrhea, many issues like that. And one of the things that we found in these mice was that the immune system of the gut is not normal. So, that's something that I'd love to explore in Wiedemann-Steiner as well, but it's like a new clue to why these kids have all these gut issues. Okay, to study whether you can have a therapy, you have to have a way to measure the mice. And we've used a test called the Morris water maze for Kabuki syndrome. It's a tank, you fill it with water, you make the water opaque, and then you teach the mice to find a platform, and they get better at it every day. And then you can say, are there differences between the mice that have the Kabuki mutation or the mice that don't have that change? And when we've done that, we found that the Kabuki mice cross in yellow. They have much fewer what is called platform crossing. So they seem to have a hippocampal memory defect. They don't learn as well as the other mice. So that's something we can measure. And then when we started to look at the other thing you can do with mice is you can look in their brain. And when we looked in their brain, we found that the number of new neurons if you look at the green signal on the left, there's a lot of these kind of cells that are labeled green, and we seem to find fewer of these cells in the Kabuki mice compared to the other mice. So they don't make as many neurons, or they're not making neurons at the same rate as the other mice. Okay, so, and this was a kind of a traumatic stumble how we got into this particular therapy, but we had worked with a drug company that had been sponsored a full trial. And I'm going to tell you about the results from this. Drug company is called Takeda. And we had this idea that KMT2D puts on this epigenetic mark, H3K4 methylation, but then there's an enzyme that removes that mark. And this drug company owned an agent that could inhibit that enzyme. So that's a very kind of obvious therapeutic possibility. And so then we've tried, you know, these are mice that have been given vehicle, so they don't have the drug, and we see a big difference in the number of the green cells. And then on the drug, which is called TAC418, we see a rescue of that measurement. So there's more of these green cells. And when we quantify this, the person is blinded and counts the amount of either this label. We can see a rescue of this measurement. And similar in this Morris water maze, we always see this abnormality in the untreated mice. But when the mice are treated with this agent called TAC418, they do better at this task. These are 3 independent tasks, but they all show the same result. So that's pretty promising. The sad thing that happened here is that the big drug company that owned this agent, they said, look, Kabuki is too rare. we're not going to work on this. But we published our paper, and we were able to reach, get in contact with this company here called Horizon. And they are working on starting the first clinical trial for that disorder. Again, I think there's a lot that can be learned from a clinical trial, and I'm hopeful it will be successful. But I just wanted you to know that this is kind of the process. You make a mouse model, you come up with a therapy, and that's the kind of stepping stone to kind of go to a clinical trial. Okay, so this is the summary for the first part. There was this clue for Kabuki syndrome. There was something wrong with opening of chromatin, putting on these histone marks. We have mice that we can model the disorder, and then we have a potential drug that could potentially be helpful in that disorder. So, one of the ideas with our clinic was that we could maybe see a lot of these disorders. And like I said, when we started, there was about 20 such disorders, but now there's 90. And this has become a large portion of intellectual disability. Like when you find a cause for intellectual disability, this is one of the biggest groups of disorders. And I think we can learn, from the different disorders to try to help the individual disorders. And when you look, I know it's a complicated figure, it's this big pie chart, but each little slice is a different gene. When you look at what they have in common, if you look at the blue color, they all have problems with intellectual disability. So that's a defining feature. There's something when you disrupt this machinery that you end up with intellectual disability. The yellow part is growth. There's also a lot of growth problems. The other thing that I wanted to address is that almost all of these have been found in cancer in one or the other way. But in general, the patients don't get cancer. That's been our experience. The only, when we look at it as a whole, the only disorders that have an elevated cancer risk are the disorders where you have overgrowth. And Wiedemann-Steiner doesn't have that there. They have small growth. So there are some disorders here where, the kids are bigger, and those kids seem to have a little bit of a cancer risk. But I don't think we think that there's a very elevated risk for any of these disorders, including Wiedemann-Steiner. But we see these themes: growth, intellectual disability. And the other weird thing that comes out of this is that this group breaks a rule in genetics. So in general, in genetics, when you have enzymes, enzymes tend to tolerate the loss of one gene. And in Wiedemann-Steiner and all these other disorders, you've lost one copy. You have another completely fine copy. So it's a little bit curious why these actually end up leading to disease, because most enzymes you need to lose both copies to get the problem. The other thing that worth for this group to know is that all of these disorders, or many of these disorders, have these diagnostic episignatures. So it's another test that can be done. So if your child has a variant that the doctor can't interpret, this is a test that can be used to help the doctor interpret the variant. It's like we see a certain signature of epigenetic marks whenever we have real cases of Wiedemann-Steiner. And then the kids that don't have that, their variant could be wrong, and maybe they have something else. So it's good for, and the test is now called Episign, and most physicians can order this now if needed. Okay, this is very sciencey, but I just wanted to mention it because of this theme of like, how can we use these disorders together to learn some fundamental truths? And one of the things we've done is we've looked at all the factors that are known that have an epigenetic function, and we've started to look at them kind of systematically. And what we find, just to tell you very briefly, is that many of these genes seem to regulate other genes in this group. So they form something called a co-expression cluster. It's shown here. We call it the highly co-expressed group. We think this might be one of the reasons why it's enough to lose one copy, because we think that these genes regulate one another. And even by losing one copy, maybe you dysregulate this big network of genes. And this is why I keep saying, I think it's useful to think about these disorders together. We can take some of the lessons from one and move it to another. Okay, and we've tested this. We've taken 3 of these disorders and asked, are some of the epigenetic marks dysregulated in the same way? And when we looked at these 3 disorders called Rubinstein-Taybi, Kabuki 1, and Kabuki 2, we did find some so more than expected by chance of the same dysregulation. So when we look at the regions, we find epigenetic abnormalities at the same locations in all 3 disorders. And we think this might be one way to kind of hone in on which parts of the genomes are most important to kind of understand the disease. Okay, so just wanted the punchline here is Wiedemann-Steiner is part of a big group of disorders. The other disorders have a lot of shared phenotypes. They also have intellectual problems, growth problems, and there seem to be shared abnormalities. These epigenetic marks are disrupted in a similar way in many of them. Okay, I have a particular interest in Kabuki, which I already told you, but also Wiedemann-Steiner, because the proteins called KMT2D or KMT2A, also called MLL or MLL2, they're very similar, and they seem to affect the same epigenetic mark. This thing called H3K4 trimethylation. So, I was kind of drawn to Wiedemann-Steiner because of that. And even when we've done sequencing to try to find, so for instance, this is a study where we were looking at people that had a diagnosis of Kabuki syndrome. And when we sequenced them, we found 2 people with Wiedemann-Steiner. So sometimes the clinicians confuse the 2 because some of the features are similar. And when we look at specific regions, that there seem to be places where the epigenetic abnormality is similar between Kabuki and Wiedemann-Steiner. For Kabuki syndrome, this work had been done, and this is all work that Tina did while she was waiting for the mice to get ready to work on them. But there was this beautiful study by a guy in the UK, his name is Sid Banka, where he took all the missense changes for all the patients with Kabuki, and he asked, do they land more frequently in one region of the gene than other, to try to figure out what is the most important part of that gene. And in his study, he found that the enzyme domain, the thing that maintains the epigenetic mark, was the most important thing. So we wanted to do this for KMT2A, the Wiedemann-Steiner gene. And I kind of expected to see the same issue, that the enzyme domain would show up. But when Tina did this, she didn't find that, and that's always good when you find something new. This protein, which you probably heard about, KMT2A, is a little bit odd because it is made in full length, but then it's cut into two pieces. There's one, it's called the N terminal and the C terminal. It forms kind of two proteins at the end, and then they work together is the idea. And Tina systematically looked at where all the mutations that are of all the patients in the literature, all the papers that have come out, where do they land? And what she found is it was a little bit in the enzyme domain. We see an enrichment there, but the biggest enrichment was in the reader domain of this protein, something called the CXXC-domain. And those two things are on the two different pieces. You know, one piece of this protein is the reader, and then the other piece is this enzyme function that puts on the epigenetic marks. So this was quite surprising to us. She looked in other genes that have CXXC domains to see if that domain is just always very enriched. And what she found, it was only for KMT2A, not for the other available proteins. When we looked at the epigenetic dysregulation, so what the CXXC domain does, it reads something called a CPG, which is, part of the which is in the kind of regulatory part of the genes. And she found that where the epigenetic abnormality in cells where they had knocked out this gene in the brain of mice, what she found is that if there was a lot of these CPGs, which is what the CXXC domain reads, that's where you saw the biggest problems. And then if you went to regions where there were fewer of these CPGs, there were less epigenetic problems. So we think this is a real finding. Another thing that Tina did, which was pretty amazing, is she used this new tool. One of the biggest breakthroughs in biology in the last couple of years is this thing called AlphaFold. And it's a way to predict the structure of proteins based on the sequence. It's a artificial intelligence type of tool. And she systematically went through all the changes that have been found in individuals with Wiedemann-Steiner. And she was able to correlate her data with experimental data that was available. And she was able to define what any change in this domain would do, whether it would be likely to lead to disease or not. And I think this could be quite useful for clinicians going forward that might want to, as new mutations come, new variants are found, sometimes it's hard to interpret them, but this work that she has done may be helpful for that. Okay, so who cares? Like, why do we need to know this? So, the reason why we're excited about this is, the KMT2A, the Wiedemann-Steiner gene, is too big for gene therapy. We've known that for a while. It's just a really, really big gene. However, if one part of this, remember this gets cut into two pieces. If one of the pieces is enough, that would be the size that could be appropriate for gene therapy. So it kind of opens up this new avenue. And again, it's not what we expected, but I started this talking about the similarities between the different conditions. But I think it's very useful to know what's specific for Wiedemann-Steiner. So this is something, and one of the things that Tin and I have been obsessed about is, can you test it in cells or in mice or something to see whether it's enough to get that a portion of the protein, not the whole protein. Would that be enough to treat the condition? Okay, just to summarize this, I know my talk is pretty sciencey, but we'll have time to chat afterwards. But we find that, to our surprise, this reader domain seems to be super important for Wiedemann-Steiner. The epigenetic abnormalities in mice are more pronounced at the locations which this reader reads. And then we've used this AlphaFold program to kind of tally all the possible changes that could happen in this region. Okay, so for future steps, you've already heard by Tinna about this specialized mouse that we're developing. And we're hopeful, if this works, that this would be a way to show that aspects of Wiedemann-Steiner can be treated. That's what we're hoping to show, because we think if we could show that, it would bring in a lot of investigators that would want to try their therapy on this condition or these mice. And obviously we'll share these mice with the community once we've made sure that they work. Like I told you, we're interested to see, and Tinna is doing some cell work to see if we can use portions of the protein or portions of the gene to fix problems in the cells or in the mice. And then after we found this, we're pretty interested to maybe look in more detail into comparing patients with mutations in that domain versus other changes. So that's one of the things that we've been thinking about. Okay, so the work I talked about is mainly Tinna's work. I basically just kind of meet with her once in a while, but it's really her that is moving this along. And these are all the people in my lab that really get the work done. I make sure there's money for them, but this is the real brain trust of my group. And I'm super thankful to them. I'm thankful to the Wiedemann-Steiner Foundation for funding us, because it's hard to get something, even if you don't have someone working on it, if you don't have a dedicated person, things move very slowly. But now that we do have someone that is dedicated to Wiedemann-Steiner, I feel like we've already made some progress, and I look forward reporting more progress in the future. So I'm not going to say any more. I'm just going to kind of open it up for questions or comments or things like that. Thank you so much.
With like the mice that you have, do you make that mouse have Kabuki?
Yeah. So yeah. No, so we make a mouse that has a change, and then we maintain that mouse. It makes other mice, and then we have those mice in the lab, and then we can compare the mice that have the change versus the mice that don't have the change. So it is amazing, but and it's really what's needed to take things to clinical trials. Like it's very hard to go from cells directly to patients, because you don't want to also do something that's risky and so forth. So with the mice, you can see whether there's any toxicity or danger of the agent. There is this other way of doing it. It's called induced pluripotent stem cells. So then they take cells from people with Wiedemann-Steiner and they transform them into a neuron or something else and study that. But my lab has always done mice with the hope that it will lead to clinical trials. So
I actually have two questions, if I can get away with two. The first question is, I love learning all the science and genetics of it, and sometimes that's just a little bit over my head. And just as an everyday parent, I wonder if you can express in succinct words, like what are the opportunities to participate in this or be a part of a part of the future of genetics. What are the opportunities that we might see in my lifetime, and what might my child at 6 years old see in her lifetime? What do you foresee for the future?
So I think for, when I started working in genetics, there were no treatments for any disorders or almost none. But the pace has really picked up, and there's a lot of amazing tools. There are small molecules, there are these gene editing strategies, and there's a lot of potential there I think, there's this institute, NHGRI, which is the genetics kind of big institute at the NIH. And they made the prediction that by 2030, there would be dozens of genetic therapies. And it's already starting to, there's a number every year where we see a new disorder with a treatment. So I'm hopeful. I don't know, but I'm hopeful there could be therapies. With therapies, they're probably going to work better the younger the child is, just to be honest. And we don't know what that window is. But I'm optimistic that there could be, in your lifetime, in her lifetime, there could be therapeutic avenues. I think this group of individuals has a lot of strengths, and I think even a minor boost could help, increase the quality of life quite a lot. So
the other aspect of my question is, when Ivy was born, I made the decision to bank her stem cells, which are still banked through cord blood storage. And I don't know, and I've sort of had this conversation before, could that be helpful down the road? And is that something, if other parents have done, would the stem cells from WSS kids be helpful to any of this?
So I don't know. I think it could be helpful for research, at least. We currently don't have a lot of examples, for brain-based disorders where stem cells have been helpful. They've been more helpful in kind of blood issues or things like that. But I don't know. It could be in the future. I don't want to say no, but right now, no. So.
And Dr. Hans, I have a question regarding the spectrum of symptoms that we see in WSS. Do you also see the same thing in Kabuki?
Yeah, there is a spectrum there, but I have the sense it's very broad in Wiedemann-Steiner compared to Kabuki. And people have done these, what we always do in clinical genetics, these genotype, phenotype, and it hasn't really led to a good explanation. My thought is there must be a modifier, or maybe a number of modifiers that are, and then if you have that modifier change, maybe you're protected or you get more severity. But because my sense, and I go to a lot of these meetings, that the spread is pretty big for Wiedemann-Steiner. So, and And I think it's something that would be worth investing in studying, because if there's a modifier that could be a treatment target, it's very hard to map modifiers, so it's not trivial. The other thing where it could be helpful is it's a little hard to give people predictions when they're diagnosed, because what are you going to say? Well, there's a big range. That's not very helpful.
Yeah.
Oh, it could be like this, or it could be like that. We'll see. So by understanding this a little bit better, I think that could be helpful to clinicians too. So.
Okay, thanks. And just an extension of that, have you seen any correlation between the severity with missense versus nonsense mutations?
Yeah, so in general, for this whole group, probably in general, the missense may be a little bit milder. I have to say, this CXXC domain, maybe there's a little bit more severity there. We'd need to study it a little bit better. And this study that Tinna kind of did as she was waiting for her mice kind of opens up that. It would be very nice to collect specifically the ones that have changes in the CXXC domain and then compare them to the rest in some way. Yeah, we don't have really good genotype phenotype correlation at this point. But the data, is getting so much better. There was just this big study that we participated in that was run through NIH CHOP that had like a number of new mutations. And then I know Wendy Jones has been for a while working on her paper. So as we have more data, maybe this will get easier to understand. But my understanding is that we don't really have a really good grasp of that, of the genotype phenotype at this point.
Thank you.
And just to piggyback off that, I think what we're doing with Rare X might be helpful because we are collecting that mutation type with the patient reported data on symptoms, uploading our genetic report. So you and Tinna could hopefully mine that for that data.
Absolutely, absolutely.
Hi there. You had spoken about a reversal drug, was it TAK?
TAK 418,
Yes.
yeah.
You said when the mice were taken off of that, there was a reversal of symptoms or of things gained is kind of what I got out of that. Am I correct in understanding that?
So when we always give the vehicle. So, whatever we mix the drug in, we give that to some of the mice just to make sure it's not that carrier that's doing something. And the mice that got the carrier only, no drug, they didn't do any better. But the ones that had the drug seemed to do better with this metric at least.
Okay. And then did you say when that's discontinued though?
We haven't done those studies.
Okay.
No, this because a lot of these mouse studies are quite cumbersome and expensive. We've only done like relatively short term, like 4 to 6 weeks or something like that. It would be really nice to do kind of long-term treatments in the mice, but we haven't done that yet.
Okay.
So
Thank you.
Yeah, I did. Hi. Thank you for being here. Just a quick question. I might not articulate myself well, but when you think about the possibility of maybe finding something in the future that might recover some of the KMT2A, a drug or whatever, is the idea that maybe you would be able to use this, and since our scale is so wide, like it would affect every kid differently, or would you have specific things that it would maybe be changing for?
No, I think that's a great question. Different kids could respond differently, right? But I think there are certain things that can be treated. The histone marks they're very dynamic and they keep changing in the brain throughout your life. So that's why we're hopeful that this could help with some of the neurological, could you make neurons maybe a little bit better or something like that. And then same with the immune system or cells that are turning over rapidly, like in the gut and stuff like that. there are other things that we could probably never treat. If there's a heart defect or some structural defect, that window is over, at birth. And the other big trend, just to mention that, is the time of diagnosis keeps going down. When I started in this field, a lot of kids were diagnosed when they were 10 or 15 or, like older. And now we see more, especially with exome sequencing, we're seeing kids getting diagnosed in the first 6 months of life. And, I think if the trend continues, maybe some of these kids are going to be diagnosed kind of at the latter part of pregnancy or something. So there may be a window of treatment if a treatment is safe and works really well. Regarding, how it's going to work on different kids, I don't know, but that's what you do clinical trials for. And my lab is kind of, we call it preclinical. I try to get things to the next level, and then people like Dr. Harris, take it that next step. And for us to be able to do clinical trials, we need to know what to measure in our kids, so that we can track whether they're improving or not. So a lot of the work that they're doing here is just to build that baseline. What are we going to measure? Can we measure it? Is it solid? I think the work is also amazingly important just for, advising families and schools and so forth, because, if we could figure out, look, this is the way that learning works for Wiedemann Steiner, maybe we could make for better education and support. So I'm sorry, I went off on a tangent.
You talked about Episign as additional testing. How do we know if our kids need that additional testing?
So it's in those cases, which is very frustrating, where the doctor says, oh, you could have Wiedemann Steiner, but I'm not sure. That's the type of kid. And that's a horrible situation to be in, because then you're like, do I belong to this group, or maybe it's something else or something? But that's the way I use this test. There's a few changes, often a lot of the changes are new in the kid. So neither parent carry them. There's some exceptions to that. So that's one way of seeing whether a change is real. But in some cases, we don't have, DNA from the parents. And then that test could be very helpful to say, oh, it's a variant we haven't seen before. We don't know if it's new or not, but the Episign says it's a real variant or it excludes it. So that's kind of the useful. And I just wanted to mention that because it is a hard position to be in if that's the case. So.
You talked about how it's in two parts, the KTM. Do you think that plays a factor in the wide variety of our symptoms of our kids? Like, do you think one is more deficit in some kids over others, and that's why they have different deficits?
Yeah, it could be. It could be the types of the mutations, where they land, and so forth. That could be one. Another possibility is it could be how much, remember, you have a full copy that's completely normal. And that's another way one can think about therapies, because if you could boost that copy, maybe you could treat the condition too. But that could also affect the severity. Like if some kids have a lot from the other copy, maybe they have a milder, or if they have less from the other copy. So that could be. And then the third possibility is that there could be other genes that are somehow affecting this, working together. And I was trying to kind of make the case that a lot of these factors seem to regulate one another. And, maybe by losing one copy, you're not just losing that gene, but you're dysregulating this whole kind of collaborative network.
If we get to the treatment point, would you have to do like specific testing to find out, since my kid doesn't have as much of one thing or the other, would you have to test to see if whatever therapy you come up with would actually be appropriate then? Just because of the variety of kids we have?
Yeah, so often if there was a clinical trial, they often would be very selective who they would pick in the beginning, and then they would expand later. So I guess the changes that would be easy to interpret or they're sure about, that would be the starting point, and then they would expand. But the hope would be to make a therapy that would work for everyone.
Just continued on that. Have you experimented with edit or CRISPR to try to take and use only the good copy, shut off the bad copy, or edit it out so we can, and then put it back in and see if it can basically reproduce itself?
Yeah, so we use this CRISPR editing all the time to make our mice, to make cell models and correct things in cell models, but we haven't looked at it as a therapeutic strategy yet. But it's a lot of these editing things work really well. They can come in and they can rescue the mutation. So now there's no problem. The difficulty is they sometimes have off-target effects, so they can affect other places. So we worry about that. And the other thing that's very difficult is it's quite a big system that needs to get into the right cells. And I think most of the CRISPR is going to be tissues that are easily accessible in the beginning. The eye, the blood cells, maybe muscles, that's where that will start. But there's great promise there. And I think this will be a viable opportunity for many disorders. And that's one of the motivations for the project that Tin and I are trying to push forward. I think if you could genetically show with that mouse model that there is malleability, there is treatability here, then I think everyone would jump in and say, oh, I'm going to take my strategy and I'm going to use Wiedemann Steiner because they've already shown that has some promise. Yeah, I don't know. There's a bunch of clinical trials, like I said, in the eyes and so forth. I don't know that. And in mice, this has been done for muscular dystrophy and a couple other conditions, and it looked pretty promising there. Again, muscle is a little bit easier to access than the brain, but there's this disorder called SMA, spinal muscular atrophy. And that's a neurological disease. There's this oligo they use there's like a mistake where some of the gene isn't put in the right format. And they've been able to inject this into the spinal cord. And that has led to very good therapy for that disorder. So yeah, there's. No, basically, so in SMA, you have two copies, one that works really well, and you have one that has a mutation, and they found a way to up the one that has the mutation. So I think there's a lot of promise there as well. I think there's a lot going to happen in the next 5 to 10 years. There's just, I think CRISPR is speeding up our progress where we can get models quicker. I think this tool that Tinna so amazingly used, AlphaFold, it's a way to understand what the mutations are doing to our proteins. So there's a lot of reasons why I think things are going to speed up, and I'm hopeful. Again, there's still a lot of work to be done, but and relatively little has been done for Wiedemann Steiner compared to other conditions. So, but I'm impressed that there's more people now coming to these meetings and working on this. So.
So we have some other questions.
Okay. No, just for everybody in here, it's just a statement that when we first met him in Baltimore, that Kabuki was we were trying to piggyback off Kabuki, but with having a pharma study, it's massive.
Sorry, I had another question, and this isn't about therapy per se, so it might not be the appropriate question for you, but I know Jillian had mentioned, I think in her testimony, one of the other ladies in here, and myself as well, in terms of like finding Wiedemann-Steiner as early as possible, like my test came back high risk for something, trisomy 13, which obviously has nothing to do with WSS. And I know she came back with high risk of Downs. Like, do you have any thoughts or anything like in terms of like, we're taking those tests, something's coming back and obviously those are negative, but is that correlated because our kids had something and it's coming back?
Yeah, we see that a lot. There's sometimes we call it like soft signs or something, that there's some flag that is raised. I think if treatments open up, I think it also lowers the threshold to test for conditions, and and also like I said, the technology to find, you don't have to, in the past you had to be pretty sophisticated to make a guess. Like for me, Wiedemann-Steiner is very easy because I've gotten the chance to meet all your kids and seen so many. But for a lot of clinicians, they may never have seen anyone and they don't think of this. But now there's this new test, the exome sequencing, that you don't have to be smart. You just have to know there's something wrong. I'm just going to see what it is, and it will give you an answer. And that's why there's an explosion in the number of cases being diagnosed. I think at some point this will move to prenatal, but it's not there yet.
Thank you for this opportunity to learn. One question going back to what you said about SMA, I was wondering, since the neurological disease does a therapy work when the child or the patient is older, or at what point is ideal to introduce a therapy?
So for SMA, we can answer this question very well. We don't know for other disorders what the windows are going to be, but for SMA, it works for everyone. But the biggest impact is at birth. If you can start the therapy at birth, you get the biggest bang for your buck. But at any time point, we've only tested up till 18 years of age. So it's currently only approved for kids, for people under 18. But I think it's probably going to work after that. There are studies to do that, but you get less impact the later you wait. So, and again, for intellectual disability, I think for a long time, people have been told that you can't do anything for intellectual disability. But I think I think we didn't know how plastic the brain is. And when I talk about plastic, let me give you an example. If you're a newborn baby and you have a malformation of the brain and they remove half of your brain, you function as a normal person, when you get older. There's no obvious or very little obvious deficits. If you take an older child and you do that, there are many deficits, but not as bad as if it's an adult. An adult that has a stroke has massive deficits and some return, but the later you are, the more deficits you get if you lose half your brain. I think what that tells us is that a child's brain is able to be plastic, and I think that's the strongest evidence that there will be some cases of intellectual disability that can be treated in childhood at least.
Hi, I found Tina's slide, or her whole presentation, so inspiring and so fascinating, and like super proud that it was funded by everybody here, by the parents. Like, we're the ones who are responsible for the fact that we know so much based on these mice. And so what is it that we can all do? I mean, like, we're the ones who are funding this. Like, tell us, like, how much do we have to raise each year? What can we do? How do we continue it? What I don't want to have happen is that we get so far and then the funding dries out. Like, that we have to make sure, like, right now it's on us to do this until we start getting these bigger, until the government grants come in and pharmacies start to donate. It's going to be us. So you tell us what you need, and then we'll figure it out, right?
So again, we're super thankful. I'm very proud of Tina, and but I think there's more work to be done, and I think you can help in many ways. One is by fundraising, because sometimes once you get the ball rolling, you can bring in bigger grants, but you often need a lot of data before you can get a governmental grant or another grant. I've been affiliated with the foundation, but this has allowed us to be a little bit much more focused in moving things forward. But the other things are also important, kind of participating in studies. And if you have thoughts for us, we're open to that as well. What you think is most important and how we should address it. These things take time. The mice don't always behave in the way, and even the best plans are sometimes quite challenging, as Tina has learned. You can have really good ideas, but at the end, it's about the execution and kind of getting it done. But we are amazingly thankful for your support, and we hope we can yield something. But I think it's a starting point. I think we can build upon this.
And we will gladly continue to fund you as best we can, because I know that time is of the essence. And kind of like John said, in 2015, 2017, when we were all together, Hans told us as a board, what you are doing is incredible, but really don't think that it's going to have an impact on your child. And in 2019, that was really a 180, where what you are doing could have an impact. Regardless, we would have kept going. But I think what we're hearing today is that there could be some big changes for our kiddos in their lifetime. And we can't thank you enough for your devotion to our community. So thank you.
Thank you.
We have let's do a 10-minute, 5-minute. Okay, we are going to take a 10-minute break, and then just a few things for the afternoon. If you're